Welcome to this Biomax video covering disease maps in the BioXM Knowledge Management Environment. In this video, we'll cover the basics about disease maps. A disease map represents one approach to organize and explore the exploding knowledge available in disease research. So let's look a bit more closely at this simple disease map for respiratory tract infections, RSV and flu. The concept of a disease map is relatively new, but basically it's a dynamic, computer-based knowledge repository that graphically represents interactions between molecular components like genes, proteins, or compounds, and pathways reported to play a role in disease pathogenesis and progression. The BioXM graph provides an ideal platform for creating, exploring, and enriching these disease maps. The disease map navigation and exploration tools help the user to visualize known associations in the context of prior knowledge about the disease. We can also enrich the disease map with any kind of information. For example, data provided by integrated public databases like Ontogene and PubChem, ontologies like Go or the NCBI taxonomy, or structured sources like KEGG pathways. Experimental data, like gene expression data, can also be visualized in the disease map to further elucidate disease-related mechanisms. But let's start at a simple level. What we've displayed here is a disease map of respiratory tract infections caused by certain types of viruses. We can click on the map to move it by drag and drop, or zoom in and out to see certain sections or details of the map. Here we see three different types of nodes in this map. In yellow, we have selected pathogens that are involved in respiratory diseases, including influenza A virus or respiratory syncytial virus, also known as RSV. These come from the NCBI taxonomy. Genes are depicted with this violet symbol, and in white boxes we have respiratory diseases like pneumonia or bronchiolitis. We also see edges between the different nodes. These represent the relations or associations of the two nodes. In this map, they're based on the results of the text mining analysis tool integrated directly in the BioXM platform. You can use the icons here in the menu bar to zoom in, zoom out, and fit. We can also display different types of information in the map by selecting the graph state here in the menu bar. This will display the specific wording which was identified in the text mining analysis and display it between the nodes. So if we focus here on the pathogen Bordetella pertussis and its relationship to TLR4, you can see the relation has positive effect. Or as it was identified in the text mining results, the exact wording was Bordetella pertussis stimulates the toll-like receptor 4 response to control infection. We also see that Bordetella pertussis has a positive effect on two diseases, pneumonia and respiratory system infectious disease. All right, we also have additional information which can be displayed in the graph, like the amount of literature evidence identified in the text mining results. We can just select the layout literature evidence, and we see the amount of literature evidence for each interaction represented by the thickness of the arrows. For instance, here we have 29 pieces of literature evidence linking RSV and respiratory system infectious diseases, or a thinner arrow which indicates that there are only 12 pieces of evidence linking it to pneumonia. You get additional information by clicking on any object, like the nodes or the interactions. For example, taking the relationship here between influenza A virus and DDX58. Here we see that DDX58 has a neutral effect on the influenza virus. If we click on the arrow, we get the text mining analysis results in a new window. Here we see information about DDX58, which is also known as RIG-I, or retinoic acid inducible gene. It's a receptor involved in sensing RNA viruses, and here the influenza A virus. Here we see that interferon beta production by influenza A virus is mediated by DDX58. And down here we have the exact sentence identified in the text mining analysis and a link to the PubMed ID. If we click on that, we get the exact abstract. And here we have further information within the abstract that RIG-I, its adapter interferon beta promoter stimulator 1, or IPS1, and interferon regulated factor 3, or IRF3, were involved in influenza A virus-mediated production of the antiviral interferon beta. This is also depicted within the graph. Let's zoom in here where we see DDX58, or Rig-I, with its adapter molecule MAVS, which is also known as IPS1, and IRF3, which have an effect on influenza A virus to produce interferon beta. All three relations here originate from that single publication in Cellular Biology from 2007. Okay, those are the basics on how to navigate a disease map. We can also further enrich such a map with new layers of information, for example, by adding a new cell type. So we have this association between interleukin-17A and RSV, where the production of interleukin-17A is induced upon infection by RSV. And interleukin-17A in turn has a positive effect on the production of LCN2. That's a protein called lipocalin-2, which is an antimicrobial iron-binding protein associated with neutrophils. Lipocalin is known to play a significant role in the acute innate immune response to infection. And the literature also shows that interleukin-17A is produced by the T helper cell. So now I would like to add such a T cell to our disease map to depict this relationship. We can do this with the graph editor, which allows us to be more interactive with the whole system. This button here opens the map in the external graph editor. So this is the same disease map, just shown in the editor, and we would now like to add a T cell to the graph. So here on the left side of the editor we have icons for displaying the cell types or compounds or proteins, which can all be added to the graph. We'll click on the cell types icon to display the table, and then we can just drag and drop one of these cell types into the graph. So we select our T cell and just drag and drop it right onto the disease map. We click OK. And now we've added this T cell to the graph. We can also add another type of cell, like neutrophils, since the production of interleukin-17A, together with other cytokines such as TNF, is known to lead to the recruitment of neutrophils into the lungs of infected patients. So we open up the cell types table again and select neutrophil this time, and then we drag it and drop it right onto the graph just like before. Here with the layout tool, we can click on any item and just move it around in the graph. And we can also add a drawing tool like a box to mark this area as involved with neutrophil recruitment. So we just click it right into the graph and add drawing tool, rounded rectangle. We click on it and enlarge it, and then we position it. We can also edit it to add the label neutrophil recruitment. Okay, that's just one way you can add information to a disease map in the BioXM platform. In the next video, we'll cover creating a disease map from scratch and enriching it with information. For further information, visit biomax.com or send us an email at bioxm@biomax.com.